In this chapter, we're learning about the basics of the cell structure. Cells were first discovered by Robert Hooke in 1665 when he was looking at cork from the cork tree. And he was using a very rudimentary kind of magnifying glass, could be called a microscope for its day. And he noticed that the little segments that made up that part of the tree, they looked like little rooms in which monks live, which are called cells. Schleiden and Schwann, quite a bit later, proposed the cell theory. All organisms are made up of cells. So in order to be a living organism, you are made up of one or more cells. Cells are the smallest living things. So the parts that make up the cell are not alive. They cannot function, duplicate, process energy on their own. So organelles, macromolecules, atoms are not alive. Viruses, which are much less than a cell, and I'll tell you a little bit more about that in this chapter, are not alive. All cells arise from pre-existing cells. In fact, all cells that are alive on Earth today can trace back their ancestry to a very first cell that formed several billion years ago. Most cells are relatively small, and this is because of the physics that limits their function. Cells must be able to allow substances to pass in and out of them in a timely manner, so in the amount of time that's sustainable for life. If it takes a year for a molecule to make it from the outside to the center of the cell's going to die before that process happens. And if the cell is really large, then the movement of substances, a lot of which happens passively by just kind of bouncing around and making its way to the right place, is very slow. Rate of diffusion, so this movement, this passive movement, it depends on the cell size, depends on temperature, concentration of substances, and distance that needs to be traveled. The most important thing is that it depends on the surface area of the cell. So just how much of the covering there is of the cell. Here, let's compare two cells. One has a radius that is 10 times larger than the other. Here, we're just calling it 1 unit. So from the center to the edge of the cell, it's 1 unit, whatever that is, maybe micrometer, whereas the other cell is 10 units. And then see how, if you were to calculate both the volume inside of each cell and the surface area, like how much of that skin surrounds, if you were to make a cell out of fabric, like how much fabric do you actually need to cover the whole surface of the cell, then volume increases 1,000 times while the surface area only increases by 100 times. So the surface area to volume ratio is 3 for the smaller cell in this example, where it's 0.3 for the larger cell in this example. You don't need to be able to calculate any of this, but I want you to just keep in mind the idea that if something just becomes bigger, when it's a sphere or a cube, a three-dimensional object that becomes bigger, the surface area and the volume don't increase in a linear proportion, and that the volume increases much faster than the surface area. Because cells need to have a lot of surface area for things to move in and out, it's better if they stay small. They function better if they stay small. Some cells are able to compensate for this by being really long and skinny, which still allows you to have a lot of surface area, but not a huge volume. So our neurons, our brain cells, they can be quite large. Some go from the bottom of our head all the way to our legs, and that's a pretty long cell. However, they're really thin, so they still have enough surface area for their volume. This is a logarithmic scale of different sizes of different things that you might be familiar with. So notice how an atom compares to a cell of a bacterial cell versus a animal cell versus a human, full-grown human. All cells of every living thing have several things in common. They all have a specifically designated area of the cell that holds all of that cell's DNA. If you remember from chapter 3, DNA is a long polymer made up of nucleotides, and in most animal cells there are many, depending on what species you are, of these long chains. They all need to be neatly stored somewhere and organized in a way, like a well-organized library, that when you need to access some information, you, the cell, then you can do so pretty easily. So this is a very important area of the cell. All cells have a cytoplasm. So cyto means having to do with the cell, and cytoplasm, the space inside of the cell. I'll come back to specifics of this, but it's a liquidy jello-like substance that's water-based with a lot of different ions, molecules, other substances in there. That whole space is called the cytoplasm. All living cells have ribosomes. Ribosomes are little machines, biological machines, that are able to make proteins. They are the makers of proteins. And remember from chapter 3 that proteins do almost everything in living cells, and they often are even the building material for a lot of cells. So it's really important that we're constantly making the necessary proteins for the cell. Ribosomes are the little substances that do it. And we will talk a lot more about ribosomes in a future chapter and today as well. How many ribosomes are in a particular cell very highly depends on the job of that cell. So if you're a cell in the pancreas, you have a lot of ribosomes because the pancreas creates a lot of proteins that get secreted into your blood and travel all over your body to be used elsewhere. However, other cells only make proteins for that particular cell, so for themselves, and therefore they have a lot less ribosomes. And the last common structure in all cells is a plasma membrane. Sometimes we refer to it as cell membrane. So plasma membrane or cell membrane, which is less accurate but often used. It is the covering of the cell. So if we imagine that a cell is a water balloon, then the actual latex that holds all the watery stuff inside would be the plasma membrane. Plasma membrane is mostly made out of phospholipids, which we learned about in chapter 3. These phospholipids are organized in a double row to create a whole sphere, with those long tails that are completely made up of hydrocarbons facing toward each other. We will learn the details of plasma membranes and other membranes in the next chapter. For now, you just need to know that the plasma membrane, cell membrane, is made out of phospholipids in all cells, with other things included for variety. And the word membrane is referring to substances that are made out of this double layer of phospholipids. And the word membrane is going to be used a lot today and in future chapters. So those four things are common to all cells in all three domains of life. However, if you remember from chapter 1, two of the three domains, bacteria and archaea, are both made up of organisms that have a particular cell type, meaning it's a whole different way from doing cells than our animal cells are made. That type of cells is called prokaryotic cells. In this case, pro means before and karyo means nucleus. Prokaryotic cells, meaning cells before nucleus, or a better way to remember, cells without nucleus. All prokaryotic organisms, so all organisms that are made of this cell type, are single-celled organisms. And they are either bacteria or archaea. If you don't know what archaea is, it's okay for now. They're similar to bacteria, but different enough to obviously have their whole different category. People used to think that archaea is just a kind of bacteria, but then we learned they're actually not at all a bacteria. But in this class, we just don't have time to explain the difference. But again, you're welcome to take my Principles of Biology class where we go a little bit more into that. Or probably even better, if you're interested in this, is microbiology, where you have a decent amount of time to discover the difference between bacteria and archaea. For now, what you need to know about prokaryotic cells, which is a cell type that is found in bacteria and archaea, is that their DNA is put to the part of a cell that's called a nucleoid region. Imagine if the cell is a studio apartment. So it's just one room, and the DNA is the computer that is standing in the corner part of your studio apartment. So there's a specific area for it, but it doesn't have walls around it. It's not a separate room. It's just an area where the computer sits. Well, in prokaryotic cells, you don't have a lot of compartmentalization. So it's like a studio apartment where you have several areas that are designated for doing different things for the cell, but nothing is completely blocked off from the rest of the cell. Prokaryotic cells, so this is all bacteria and all archaea, they have their DNA in a specific area of the cell, but it's not in a separate compartment. And that area is called the nucleoid region. Outside of their plasma membrane, they also have a hard-walled container that we call the cell wall. Prokaryotic cells have cell walls outside of their plasma membrane. And all of these prokaryotic cell types, they have ribosomes. So those little machines, biological machines that make proteins, they have them just as we have them. They're not identical, but they're really close to being the same. Here's an electron micrograph as well as a drawing of a prokaryotic cell. The thread-like blue thing is representing the long polymer of DNA, and it's in a particular area. In this picture, it looks like it's taking up the whole cell, but it's in a particular area. And the little dots here are representing ribosomes, or those little biological protein makers. There are several other structures that differ between different kind of prokaryotic organisms. This one has a flagellum, which is that long tail. It also has cilia, which is like the little hairs, and all of these things are possible, but not always present in bacteria and archaea. All we're going to say about prokaryotic cells, although they're crucial for the health of our planet and for our own health as a human being. In fact, if you were to count all the cells in and on you right now, more of them by count would be prokaryotic cells, so cells of bacteria and archaea that are inside you and on you and in every part of you, than actual like animal cells that you're made up of. We're mostly made up of bacteria and archaea. And this is a really good thing. Many of them do hugely beneficial things for our body, and we would not be able to survive without them. And the rest of them are largely neutral. But neutral is actually good because they cover all of us. so that there's no room for something harmful to come and find a place. Like, ooh, this is a great place to live and there's a lot of food I can eat here, so I'm going to live here, and because of all the great living arrangements, I'm going to multiply and create a huge population of these disease-causing bacteria. If you have a healthy microbiome, so covered with all the good and neutral bacteria, you're way less likely to get colonized by something that is disease causing. So we rely on those archaean bacteria to keep us healthy. Now we will spend the rest of the chapter talking about eukaryotic cells. Cells in the third domain of life, called eukarya, all have a different cell type, and that cell type is called eukaryotic cells. Eukaryotic is true nucleus. So for karyo being nucleus. Remember, prokaryotic means before nucleus or without a nucleus. Eukaryotic means a true nucleus or with a nucleus. So what the heck is a nucleus? Well, all eukaryotic cells, they have a special compartment to store all DNA of that cell. So what that means is those long chains of nucleotides that code for information in our cells, they are very carefully stored in like an extra bubble. It's another compartment inside of a cell that's usually spherical. Its main job is to protect and keep organized all the long polymers of DNA that we have. And that compartment inside of our cell is called a nucleus. I know it's confusing because we talked about the nucleus of the atom being a center of the atom where you have protons and neutrons. And this is a nucleus of a cell. But they have nothing to do with one another. If a prokaryotic cell was like a studio apartment where it's all one room, but there's areas for different jobs, like this is where you sleep because the bed is here. This is where you work because the computer's here. Then a eukaryotic cell can be thought of as a full-blown house that has separate rooms that have walls completely separating the different areas. Eukaryotic cells also have cytoskeleton. So cyto means cell and skeleton is skeleton. So like a skeleton inside of a cell. It's not made out of bones, it's made out of strong proteins to hold the shape of a cell and guide the movement inside of that cell. And eukaryotic cells have other compartments which we call organelles, where you can have a very specific environment. It's really helpful because imagine that you're in your house and you want to make chocolate chip cookies. To achieve a nice crunch on the outside but gooey on the inside of a chocolate chip cookie, you have to caramelize the sugar. So you have to turn your oven up to 375 degrees Fahrenheit, and you're really glad that you have an oven that is its own separate compartment where you can put in your dough, turn on the really high temperature, and let something specific that you need happen. If you didn't have that compartment, but you really wanted to make that cookie, you would have to heat up your entire house of residence to that high temperature, and that would burn and ruin everything. So that's not possible, right? It's really nice to have several compartments that can have very unique environments that can have unique chemical processes happening without interfering or ruining stuff in other areas. And this is why compartmentalization, having different sections that are completely blocked off in eukaryotic cells, is helpful for the kind of life eukaryotic cells lead. Here's a cartoon picture of a generic animal cell. It's a cross section. It's colored in order to help you distinguish different features, but in reality, these parts of our cell do not have these bright colors. And also everything is way more smushed and squished together in an actual cell than is shown here. This is a plant cell. It's very similar to an animal cell, but has a few distinguishing features. All plants, animals, and fungi are eukaryotes, so they have a eukaryotic or compartmentalized cell type. There are other organisms that are eukaryotes that don't fit into plant, animal, and fungi, and we call them protists. Okay, so plants, animals, fungi, and protists are all eukaryotic organisms because they have a complete nucleus, a full compartment that holds the DNA, and they have other organelles in order to have specified compartmentalization in the cell. Let's look a little bit at the nucleus because it's quite a complex organelle. The nucleus is made up of two double layers of phospholipids. This is the actual outside of the nucleus. Again, we will learn more about these membranes in the next chapter. So it has two membranes that are made up of double layers of phospholipids. If you don't remember what phospholipids are, go to the end of chapter 3 right now and look at them and how they form membranes so we can continue on. Inside of that sphere of two double layers of phospholipids, there are pores. They're almost like holes made out of proteins, but they're not holes. They are a protein with a tunnel inside. And this is important because although you want to keep everything in the nucleus really protected, we still need some controlled movement in and out of the nucleus. Also inside of the nucleus, there's a region, so not a compartment, but a region called the nucleolus, where ribosomes, or those biological machines that make proteins, ribosomes are made in the nucleolus, which is in the nucleus. Confusing because the words are really similar, but write it down in your notes. Nucleus is a spherical compartment, is made up of two phospholipid double layers. It has protein pores through it to allow movement in and out of this compartment. And inside the nucleus, there is an area, not a compartment, but an area where ribosomes are made, and it's called the nucleolus. The DNA in eukaryotic cells is made out of several long polymers of DNA nucleotides. So it's not just one polymer of DNA and that's your code. In humans, there are actually 46 of these long chains of DNA polymer that code all the information needed for the cell. The exception for humans is your gametes or sex cells, so the egg and sperm, and they only have 23 of these long polymers of DNA. All your other cells have 46, if we're not going into details, because there's some differences that can exist. Go ahead and look over the drawing of all of these parts and out loud say, what is the function of nucleus in general? The nuclear envelope, or the two double layers of phospholipids that holds the whole thing together. Nuclear pores, nucleolus, and the DNA is organized into chromosomes. I'm not going to ask you about that right now. We will learn what the difference is between chromosomes and DNA in a future chapter. So just now know that there is DNA in the nucleus in several polypeptide chains. Ribosomes are present in every cell, not just in eukaryotic, but prokaryotic cells like bacteria and archaea, they have ribosomes as well. Ribosomes are not technically called organelles because they do not have a membrane, so they don't have that covering of phospholipid bilayer. They are made of a lot of different proteins clumped together. And there's also RNA clumped in there as well. So I'm going to give you a really brief description of ribosome structure. Bunch of proteins that are in their 3D blobs clumped together, and some RNA, which is a polymer of nucleotides, also clumped in there. And then you have, that's one part of ribosome, and it's called the large subunit. And then there's a small subunit of ribosomes, which is also made up of clumps of different proteins and some RNA stuck in there. So you have these two sections, both made up of proteins and RNA, and they are made separately. But when they are ready to work, like we're ready to turn on and do our job, the two clumps, the bigger subunit and the smaller subunit, stick together and they are ready to make proteins. So the primary function of ribosomes in any cell that exists is to work at building proteins. Remember, proteins are made out of amino acids linked together. Ribosomes are where this linking of amino acids happens, and they make sure that the amino acids are linked in the right order and everything is the way it should be. In eukaryotic cells, like the cells that we have and plants and fungi and other animals have, the ribosomes are, the two pieces are made in the nucleolus, which is in the nucleus. They come out using those little tunnels, nuclear pores, into the cytoplasm, which is the watery area outside of the nucleus, but still inside the cell. And they float around and produce proteins. Or they kind of park themselves, if you can imagine them being a little spaceship, they park themselves on a membrane of a different part of the cell and do their protein building there. We will talk about why they would do that instead of just floating around like a free spirit in a few minutes. So we say there are free ribosomes, those are just the float around in the watery environment of the cytoplasm, or there are bound ribosomes, those that park. They don't park there forever. Sometimes they park and then they become free later for a different job. Let's call that good for part 1. We'll talk a lot more about parts of the cell in part 2.